The broadcast is now starting. All attendees are in listen-only mode.
So, good evening from Munich. My name is Christian Zinser, and I will show you an overview of the new features in the December 2017 release of the Genomatix software suite. First, a few introductory slides, and then a live demonstration of some of the features. First of all, new data background. We have two new genomes, one for the chimpanzee and one for the Western clawed frog. We have new transcript annotation for all 33 genomes in the system. And we have new literature now based on PubMed from November 2017. And we also have new transcription factor binding site definitions in our matrix library, notably 330 new ones from plant and also 222 from vertebrate, summing up to over 2,000 binding site definitions, which we now have in our system. And for the promoter modules, so the combinations of binding sites that have been found to be functional in some contexts, we now have 30 new modules, 24 vertebrate, 6 plant, summing up to over 1,000 modules, which we have now in our system. A new features about promoter sets is that more promoters are now found in promoter sets. So the algorithm has been updated. So you get more promoters in promoter sets for the analysis of evolutionary conservation of transcription factor binding sites in your promoters. Also in the promoters, you now find that you get the top 5 tissues where the promoter is active shown in your promoter lists. This makes it easier to select the promoters which are the strongest ones in your gene if you have alternative promoters, or are the strongest ones in a specific tissue and alternative promoters. This information is available everywhere where you can select promoters from a list for subsequent analysis, which is the case in MatInspector when you do a gene-based analysis of promoters in Gene2Promoter, where you have promoters of a number of genes that you can then compare to one another, and also in the alternative transcripts view in Eldorado for a specific gene, and the comparative genomics view in Eldorado for a gene, where you also get the orthologous genes and also the orthologous promoters. We also have more additional lines of evidence for the analysis of transcription factor binding sites. On the one hand, in MatInspector, which many of you might already have used, you now also get ChIP-seq peak overlap information for mouse promoters. Up to now, only the human promoters had this kind of information, but now also in mouse, as you see here for the mouse IL-10 promoter. In Frameworker, which is our program for finding common patterns of transcription factor binding sites in a set of promoters, or generally in a set of sequences, which can form a part of regulatory modules. You now also have the additional lines of evidence, which you so far only had in MatInspector. What's only present in Frameworker actually are transcription factor interactions. This is shown here in a screenshot where we have a pattern that consists of these two binding sites, ETSF and YY1F. And we find that the matching transcription factors, for example here, ETV4 binding to the ETSF factor binding site and YY1 binding to the YY1F binding site, can actually interact with one another. So this information is taken from the literature as an additional line of evidence that this pattern is not just some random hit in a comparative analysis of promoters, but is supported by information that we have here based on the knowledge about the binding proteins. And similar to what we have in MatInspector, you also get information about whether such a module is similar to a module that we already know and that we have in our promoter module library based on literature again. So functional modules. You have information about whether the binding sites overlap with evolutionary constrained elements in human. So these elements are short sequences which are conserved in evolution. So an additional line of evidence that something is conserved here. And you also have information about ENCODE ChIP-seq data overlap. So information that specific transcription factors which match the binding sites here actually bind in that region as well. For publications, you can now export graphical views in Matinspector and Frameworker. You see a Matinspector view here. Also in the scalable vector graphics, in addition to the PNG and the JPGs, which you could already export in earlier versions of the program. This is just to show you the similar thing here in Frameworker. Then we have a few things here in Mathbase, so our transcription factor knowledge base. On the one hand, if you browse the Mathbase for transcription factors or also for promoter modules, you can now select to get only those transcription factors or modules which have been described in a specific or a number of selectable species that you have here in this list. And the second thing is that the list that you get from this analysis can now be saved as an Excel file or as a tab-separated file. You see it down here. And lastly, a new addition to the Genomatix pathway system. You might be aware of the fact that in the Genomatix pathway system, you can extend gene networks by transcription factors or also by transcriptional downstream targets and a number of other things. What we now have new is that you can filter your extensions for transcription factors or transcriptional downstream targets so that only those connected genes are shown which have either a predicted or a validated transcription factor binding site, as you see it here. Okay, now a few examples. I already have the main menu of the Genomatix software suite open here, and the first example I'd like to show you is the promoter sets and the tissue expression that you now see in the promoters and the additional lines of evidence that you can show in Frameworker. Okay, so for this I start with a gene that I get here using our Eldorado genome annotation. I use the human CDK5. And in the comparative genomics, which I like to use for Frameworker, for comparative Frameworker analysis, I now get the additional information for the tissues. You see that we have promoter sets here. We have the human gene, we have the promoters, actually 3 of them in the human gene, and we have a list of the transcripts which are regulated by these promoters. So these 2 promoters regulate 1 transcript each, and this promoter regulates a number of transcripts. And we see this in the same way for the other vertebrate species. Here, for example, the rhesus monkey and the chimpanzee and the mouse. We see that these 2 promoters are in promoter sets. This one here in promoter set 1, this in promoter set 3. In the previous versions, this promoter was not in any promoter set. So this is due to the new algorithm that does its job better to actually find the promoter sets. And you also see that this promoter E is a very relatively strong promoter. If you compare it to the other ones, you see a measure actually that's normalized the value based on cage tags, a measure for the usage of this promoter in TPM, so tags per million tags in different tissues. The occipital pole here is at number 1 with 31, and compared to that we have 0.4 and 0.055 here in the top tissues for the other 2 promoters. So this is the strongest one, and so occipital pole is actually a part of the brain. It's the end of the occipital lobe in the cerebrum, which is at the back of the head, and functionally it contains a part of the primary visual cortex. So this is a strong promoter here in the brain, and it's in promoter set 3. And we can use this if we are specifically interested in the main promoter, or specifically interested in a promoter that is strong here in this part of the brain, to run a comparative analysis, for example, with Frameworker, which I will show you, using this promoter and the other promoters in the promoter set 3 that we have in other species, like what you see here, also in promoter set 3 or here. And we can easily select the promoters just by one click. If we scroll down here in the list and select the promoter set 3, and now you see the check marks here in these check boxes, and you see that all of the promoter set 3 promoters are actually selected for downstream analysis or also for saving them. And for downstream analysis, I will use the Frameworker. As I said, this is our program for finding common patterns of transcription factor binding sites. So I run this, and here I can select the basic parameters for what type of binding sites we're actually looking for. I just leave this at the defaults. And in the next, I can set stringency parameters. I'll change this to 11 out of 13. So this would be a quorum constraint. We need to find a common pattern in 11 of our 13 input sequences. And we want to have the human sequence in our set of sequences in any case. So the pattern needs to be present in the human sequence. Okay, then we can start the Frameworker. And we find that we have 6 different patterns that consist of 2 elements, of 2 transcription factor binding sites. If we click on this link, then we see the list of these patterns consisting of the binding sites that are listed here with a strand orientation and some matrix similarity information, so how good the fit is in the end, and the distance to the next element. So this is what the pattern is actually defined by. And we find here, which is new, 4 buttons under each definition where we can show additional lines of evidence. Let's click on the first here, transcription factor interactions. Here we find that factors that can bind the binding sites here, for example, here the SP1 from human can bind the SP1F binding site, and the CBTB7A from human can bind this ZF02 binding site. They actually can interact with one another. This is information from the literature, and you can also click on this link where you get the corresponding sentences that tell you that there is interaction between the 2 genes, and you can also go to PubMed using the link then on this page. First an overview, and then the actual PubMed page with the abstract. So you can directly drill down into the information and get more details about that. Okay, so these are the interactions which show you that not only do we have a pattern here that is common in 11 sequences actually, but we also have the corresponding the corresponding factors which can interact with one another. Then model matches, in this case, there is no information, so you would get something if we had a known pattern of binding sites in our model library of functional modules. So then in this case, this would mean that it could, as it has the same or at least a similar pattern as a functional module, would be another line of evidence that this could also be functional in this context. For the constrained elements, we find, okay, in the human promoter, there is also sequence conservation in that region, giving us an additional lines of evidence that there is evolutionary conservation here. And for the ChIP-seq peak overlap, we find that two factors that can bind SP1, SP1F, either the SP1 or the SP2 factor, have been shown to actually bind using ChIP-seq peak evidence in that region and overlapping with a binding site. The same is true for the CBTB7A for the ZF02 binding site. Here are the tissues where we have the binding of these factors actually in that region, and the promoter. So this would all be in the human promoter. Okay, so much for the additional lines of evidence. Just quickly, the graphical view. Here now have the SVG download option. So if you want to see this first model, we can select the first model here and the corresponding matches that we have in the different promoters, and we can save that in as an SVG here and either download it or open it in a browser. For example, here in Google Chrome, which will then look like this, as you see it here. Okay, so much for the frameworker. Then in Math Inspector, I mentioned that you also have more additional lines of evidence now for the mouse. Just showing you that if we go to the Math Inspector, and search for a gene, I use the IL-10 gene from mouse. We again get the list of promoters which are known for the gene, and you also get again the tissue information here, and this promoter is selected. We can continue here and run the analysis. I just include the promoter module evidence. So by default, we see the experimental evidence, the literature evidence, and if you want to, then also the promoter module evidence. Run the query. Which will then, after, well, a few seconds, give us the list of the matches. Here we are. So we get the matches in Math Inspector sorted by the position. You see the matrix family here, a short description, the corresponding matrix which has the highest score, which you see here, highest matrix similarity in the family, the position information, strand information. And here we have the lines of evidence. And if you want to filter for the presence of ChIP sequencing overlap evidence, you just type something like ChIP here in this filter field, and you see that we have now with a gray background, we have a correlation with matching ChIP-seq peak. So for example, here the IRF1 factor, which can bind the IRFF binding site, which we have here, correlates, overlaps with the binding site, or down here we have an NF-kappa B binding site, and we find that the rel-A, which can bind this kind of binding site, also has a peak overlapping. And you also see, by the way, that there is also a model match with 2 of the known transcription factor binding site models that we have in our database. And same as for the frameworker, if you go to the graphical view, you can now do a scalable vector graphics export of this graph. Okay, the next thing I'd like to show you is Mathbase. Now this additional option for filtering the output, you find Mathbase here under gene regulation. And you know that you can search for specific binding sites and matrix families, etc. here, but you can also browse to see an overview. And if I now browse for, or I actually select the transcription factors I'd like to see, I can now restrict my output to 1 or more species that I have here. So I just select the cow here and click on browse for showing me only the cow transcription factors. Which gives me the list of transcription factors with a short description, synonyms, and well, the organism here certainly, and information about the binding site matrix, or in the case that it's a cofactor, you see it also here, for example. Quite a long list. And if you want to save it now, you just click on one of these 2 buttons. For example, here is an Excel file, and opening it in Excel, you get just the same information in a simple list, a simple table here. Okay, the last thing I'd like to show you today is the Genomatix pathway system and the extension options. So going back to the top here, we find the Genomatix pathway system here under literature and pathways in the menu bar. And you can run characterization of gene sets here. I'll just show you an example here with our example gene set, which I already have opened here. So we don't need to wait for it to load. And you see here on the left-hand side, in this box, we have the different overrepresented biological categories or terms in different biological categories. You see here, for example, the pathway-based networks, and the most overrepresented ones are here, starting from the top with the smallest p-values, immune system, pathway, innate immune system, etc. And I just want to pick out one here, the IL-12 mediated signaling events. 22 of the genes that are in this example set actually fall into this category to be part of this IL-12 mediated signaling events pathway. We see a network which is based now not on the pathway where we took the genes from. It's based on co-citations actually between the genes. And you see by default we have the gene nodes here in different colors for upregulation in red and downregulation in blue, as we have a differential expression data set here. And we have the cellular compartments of the different genes, or actually the gene products where they are to be found. And all these connections here show us co-citations with some additional information. You see the different arrowheads in different colors, different shapes. You see the lines, solid lines, dashed lines. You might also see a dotted lines on here, for example. And we can change the layout, which I just do here by clicking on one of the other layout buttons here. I will show you the hierarchical layout here. The program tries to arrange the different genes so that we have kind of an information flow from the top to the bottom. This is based on the information that the program can deduce from the connections and the arrows that we have in here. Now, in order to extend such a network by genes that might not fall into this category, and which might have some regulatory function or actually be regulated by genes that we have in the network, we have a number of options down here for the network extension. So we can extend just by co-cited genes that have a lot of co-citations with a gene that we already have in the network, or here for transcription factors that could regulate the gene, microRNAs that might also either regulate a gene or be regulated by that gene, and transcriptional downstream targets of a gene. Specifically, this is interesting if you have a transcription factor in your network, like you have here, for example, a STAT5A is a transcription factor. We have a known binding site, which is shown by the sequence logo just below the node. And if we select this node now and say we want to see transcriptional downstream targets, we get such a dialog here, which where we can select a few things. By default, 10 genes, up to 10 genes will be added to the network, and we can now select whether the binding sites that we need to find in the promoter of these genes need to be predicted binding sites or whether they need to be validated binding sites. So predicted would be an in silico prediction based on the sequence analysis and based on our Mathbase transcription factor binding site library, and the validated binding sites, they would be based on ChIP-seq peaks of the corresponding factor, or in this case of the STAT5A, which can actually bind or which actually binds in the promoter of the connected genes. Now, if we run this with predicted transcription factor binding sites, the program searches the database and adds the genes now with a gray background instead of a colored one, because there is no information as of now about the up or down regulation. They are not necessarily part of our input set. They can be just co-citation based connections and just from the literature. We find that we have some genes up here. And if we zoom in a bit, we see that all of these connected genes have either a gray arrowhead in different shapes or a black arrowhead actually pointing to them. Gray arrowhead signifies that we have an in silico match for a binding site for the STAT5A, here for example in the STAT5B promoter. Black means we also have a validated binding site based on chip sequencing data, but they will also have an in silico match, and this is shown here. So some of the genes are automatically arranged above the STAT5A based on the direction here, or the main direction actually of the arrows that we have here, and the other ones are placed below the STAT5A. Now, if we remove these connected genes once more, which we can do using the button down here for the network processing. This removes any genes that are not in the category that we have here. So this includes our connected genes, and run the same analysis for STAT5A now with the validated transcription factor binding sites instead of the predicted ones. Now, all of our genes are actually placed below the STAT5A. That means that the program now can deduce from the information that we have that the flow of information should go from STAT5A to the other ones. And all of them now have a validated binding of the factor in their promoters, as you see them here. Okay, now for the upstream factors, so transcription factors which could actually regulate one of the genes, just remove the others again and select MapK14 out of our network here, and now select the second option here, extension by transcription factors. And here we actually have three different options, so we can get the transcription factors with any kind of transcription factor binding site, or specifically with predicted ones, or specifically with validated ones. What do we get if we extend our network by any factor with any type of binding site? Zooming in here a bit again, we find that, well, the majority of our transcription factors actually have a known binding site, as you see here. But one of them, the PML, which is a cofactor, the cofactor nature of PML is also shown by the star shape of the node and by the absence actually of the sequence logo here. This cofactor obviously binds in the promoter of MapK14 as shown by this black arrowhead, and this is also included in our network here. You see that some of the other transcription factors have just an in silico binding site, and others have a validated binding site. And this, well, can have only a validated binding site because we have no definition for an in silico binding site for this factor. Now, if we remove the nodes once more and run the analysis with the predicted binding sites, using the second option, now the PML is gone, and all of the connected binding sites, all of the connected factors now have a known binding site. And they may have, in addition to the predicted one, they may also have a validated one, but they will have a predicted one in the MapK14 promoter. And the last way to do this now for the transcription factors with a validated binding site. Now, by the way, you also see that, again, we have the majority of our transcription factors are now above MapK14. CEPD, which we have here, is below. So this is also, again, indication that the program thinks that these guys up here should be actually upstream of MapK14. And they also all have a validated binding site as you see it here. We have our PML back again, and we also have this histone deacetylase, which also binds here, but which is a factor without a known specific sequence where it binds. Okay, so that's what I wanted to show you today. And yeah, see, no questions at the moment. Okay, so I'd like to thank you for your attention and wish you a nice day. And well, until next time, goodbye.